Hello everyone, how are you all today? I wasn't able to make any videos for a long while, but now I'm back, so it will be a fresh start for me. In this video, I will explain evolutionary biology and how it is the best explanation for how species change over time and adapt to their environment. So let's dive in. What is evolutionary biology? Evolutionary biology is a subfield of biology that studies the evolutionary processes, natural selection, common descent, and specification that produced the diversity of life on Earth. It is also defined as the study of the history of life forms on Earth. Evolution holds that all species are related and gradually change over generations. One in a population, the genetic variations affect the phenotypes, physical characteristics of an organism. These changes in the phenotypes will be an advantage to some organisms, which will then be passed on to their offspring. Some examples of evolution in species over many generations are the peppered moth and flightless birds. In the 1930s, the discipline of evolutionary biology emerged through what Julian Huxley called the modern synthesis of understanding from previously unrelated fields of biological research, such as genetics and ecology, systematics, and paleontology. The investigational range of current research has widened to encompass the genetic architecture of adaptation, molecular evolution, and the different forces that contribute to evolution, such as sexual selection, genetic drift, and biogeography. Moreover, the newer field of evolutionary developmental biology, Evo investigates how embryogenesis is controlled, thus yielding a wider synthesis that integrates developmental biology with the fields of study covered by the earlier evolutionary synthesis. Subfields evolution is the central unifying concept in biology. Biology can be divided into various ways. One way is by the level of biological organization, from molecular to cell organism to population. Another way is by perceived taxonomic group with fields such as zoology, botany, and microbiology, reflecting what was once seen as the major divisions of life. A third way is by approaches such as field biology, theoretical biology, experimental evolution, and paleontology. These alternative ways of dividing up the subject have been combined with evolutionary biology to create subfields like evolutionary ecology and evolutionary developmental biology. More recently, the merge between biological science and applied sciences gave birth to new fields that are extensions of evolutionary biology, including evolutionary robotics, engineering, 2, algorithms, 3, economics, 4, and architecture. 5. The basic mechanisms of evolution are applied directly or indirectly to come up with novel designs or solve problems that are difficult to solve otherwise. The research generated in these applied fields contribute towards progress, especially from work on evolution in computer science and engineering fields such as mechanical engineering. Different types of evolutions. Adaptive evolution relates to evolutionary changes that happen due to the changes in the environment. This makes the organism suitable to its habitat. This change increases the chances of survival and reproduction of the organism. This can be referred to as an organism's fitness. For example, Darwin's finches on Galapagos Island developed differently shaped beaks to survive for a long time. Adaptive evolution can also be convergent if two distantly related species live in similar environments facing similar pressures. Convergent evolution is the process in which related or distantly related organisms evolve identical characteristics independently. This type of evolution creates analogous structures which have a similar function, structure, or form between the two species. For example, sharks and dolphins look alike, but they are not related. Likewise, birds, flying insects, and bats can all fly, but they are unrelated. These similar traits tend to evolve from having similar environmental pressures. Divergent evolution is the process of speciation. This can occur in several ways. Allopatric speciation is when species are separated by a physical barrier that separates the population into two groups. Evolutionary mechanisms such as genetic drift and natural selection can then act independently on each population. Parapatric speciation is a type of allopatric speciation that occurs when one of the new populations is considerably smaller than the other initial population. This leads to the founder's effect, and the population can have different allele frequencies and phenotypes than the original population. These small populations are also more likely to see effects from genetic drift. Parapatric speciation is allopatric speciation but occurs when the species diverge without a physical barrier separating the population. This tends to occur when a population of a species is incredibly large and occupies a vast environment, Sympatric speciation is when a new species or subspecies sprouts from the original population while still occupying the same small environment and without any physical barriers separating them from members of their original population. There is scientific debate as to whether sympatric speciation actually exists. Artificial speciation is when scientists purposefully cause new species to emerge to use in laboratory procedures. Coevolution refers to the influence of two closely associated species. When two or more species evolve together, one species adapts to changes in the other species. This type of evolution often occurs in species that have symbiotic relationships. For example, predator-prey coevolution is the most common type, where the predator must evolve to become a more effective hunter due to selective pressure on the prey to avoid capture. This in turn leads the prey to develop better survival strategies. The red-queen hypothesis is an example of predator-prey interactions. Other common examples include the relationship between pollinating insects like bees and flowering plants, as well as herbivores and plants, which demonstrate diffuse or guild coevolution. Mechanism. The process of evolution. The mechanisms of evolution focus mainly on mutation, genetic drift, gene flow, non-random mating, and natural selection. Mutation. Mutation is a change in the DNA sequence inside a gene or a chromosome of an organism. Most mutations are deleterious or neutral, i.e., they can neither harm nor benefit, but can also be beneficial sometimes. Genetic drift. Genetic drift is a variational process. It happens as a result of the sampling errors from one generation to another generation, where a random event that happens by chance in nature changes or influences allele frequency within a population. It has a much stronger effect on small populations than large ones. Gene flow. Gene flow is the transfer of genetic material from the gene pool of one population to another. In a population, migration occurs from one species to another, resulting in the change of allele frequency. Natural selection. The survival and reproductive rate of a species depends on the adaptability of the species to their environment. This process is called natural selection. Some species with certain traits in a population have higher survival and reproductive rate than others, fitness, and they pass on these genetic features to their offspring. In evolutionary developmental biology, scientists look at how the different processes in development play a role in how a specific organism reaches its current body plan. The genetic regulation of ontogeny and the phylogenetic process is what allows for this kind of understanding of biology to be possible. By looking at different processes during development and going through the evolutionary tree, one can determine at which point a specific structure came about. For example, the three germ layers can be observed to not be present in cnidarians and ctenophores, which instead present in worms, being more or less developed depending on the kind of worm itself. Other structures like the development of Hox genes and sensory organs such as eyes can also be traced with this practice. Phylogenetic tree. Phylogenetic trees are representations of genetic lineage. They are figures that show how related species are to one another. They formed by analyzing the physical traits as well as the similarities of the DNA between species. Then by using a molecular clock, scientists can estimate when the species diverged. An example of a phylogeny would be the tree of life. Homologs, genes that have shared ancestry, are homologs. If a speciation event occurs and one gene ends up in two different species, the genes are now orthologous. If a gene is duplicated within a singular species, then it is a paralog. A molecular clock can be used to estimate when these events occurred. Evidence for evolution comes from many different areas of biology. Anatomy. Species may share similar physical features because the feature was present in a common ancestor. Homologous structures. Molecular biology. DNA and the genetic code reflect the shared ancestry of life. DNA comparisons can show how related species are. Biogeography. The global distribution of organisms and the unique features of island species reflect evolution and geological change. Fossils. Fossils document the existence of now extinct past species that are related to present-day species. Direct observation. We can directly observe small-scale evolution in organisms with short life cycles, e.g., pesticide-resistant insect. Darwin thought of evolution as if descent with modification, a process in which species change and give rise to new species over many generations. He proposed that the evolutionary history of life forms a branching tree with many levels in which all species can be traced back to an ancient common ancestor. In this tree model, more closely related groups of species have more recent common ancestors, and each group will tend to share features that were present in its last common ancestor. We can use this idea to work backwards and figure out how organisms are related based on their shared features. Homologous features: if two or more species share a unique physical feature, such as a complex bone structure or a body plan, they may all have inherited this feature from a common ancestor. Physical features shared due to evolutionary history, a common ancestor, are said to be homologous. To give one classic example, the forelimbs of whales, humans, birds, and dogs look pretty different on the outside. That's because they're adapted to function in different environments. However, if you look at the bone structure of the forelimbs, you'll find that the pattern of bones is very similar across species. It's unlikely that such similar structures would have evolved independently in each species, and more likely that the basic layout of bones was already present in a common ancestor of whales, humans, dogs, and birds. Some homologous structures can be seen only in embryos. For instance, all vertebrate embryos, including humans, have gill slits and a tail during early development. The developmental patterns of these species become more different later on, which is why your embryonic tail is now your tailbone and your gill slits have turned into your jaw and inner ear. Homologous embryonic structures reflect that the developmental programs of vertebrates are variations on a similar plan that existed in their last common ancestor. Sometimes organisms have structures that are homologous to important structures in other organisms, but that have lost their major ancestral function. These structures, which are often reduced in size, are known as vestigial structures. Examples of vestigial structures include the tailbone of humans, a vestigial tail, the hind leg bones of whales, and the underdeveloped legs found in some snakes. See picture of rat. Analogous features to make things a little more interesting and complicated. Not all physical features that look alike are marks of common ancestry. Instead, some physical similarities are analogous. They evolved independently in different organisms because the organisms lived in similar environments or experienced similar selective pressures. This process is called convergent evolution. To converge means to come together, like two lines meeting at a point. For example, two distantly related species that live in the Arctic fox and the ptarmigan, a bird, both undergo seasonal changes of color from dark to snowy white. This shared feature doesn't reflect common ancestry, i.e., it's unlikely that the last common ancestor of the fox and ptarmigan changed color with the seasons instead. This feature was favored separately in both species due to similar selective pressures. That is, the genetically determined ability to switch to light coloration in winter helped both foxes and ptarmigans survive and reproduce in a place with snowy winters and sharp-eyed predators. Determining relationships from similar features in general, biologists don't conclude how species are related based on any single feature they think is homologous. Instead, they study a large collection of features, often both physical features and DNA sequences, and conclude relatedness based on these features as a group. We will explore this idea further when we examine phylogenetic trees, a molecular biology like structural homologies. Similarities between biological molecules can reflect shared evolutionary ancestry. At the most basic level, all living organisms share the same genetic material, DNA, the same or highly similar genetic codes, the same basic process of gene expression, transcription, and translation, the same molecular building blocks, such as amino acids. These shared features suggest that all living things are descended from a common ancestor and that this ancestor had DNA as its genetic material, used the genetic code, and expressed its genes by transcription and translation. Present-day organisms all share these features because they were inherited from the ancestor and because any big changes in this basic machinery would have broken the basic functionality of cells. Although they're great for establishing the common origins of life, features like having DNA or carrying out transcription and translation are not so useful for figuring out how related particular organisms are. If we want to determine which organisms in a group are most closely related, we need to use different types of molecular features, such as the nucleotide sequences of genes. Homogeneous genes: biologists often compare the sequences of related genes found in different species, often called homologous or orthologous genes, to figure out how those species are evolutionarily related to one another. The basic idea behind this approach is that two species have the same gene because they inherited it from a common ancestor. For instance, humans, cows, chickens, and chimpanzees all have a gene that encodes the hormone insulin because this gene was already present in their last common ancestor. In general, the more DNA differences in homologous genes or amino acid differences in the proteins they encode between two species, the more distantly the species are related. For instance, human and chimpanzee insulin proteins are much more similar, about 98% identical, and human and chicken insulin proteins about 64% identical, reflecting that humans and chimpanzees are more closely related than humans and chickens. Biogeography, the geographic distribution of organisms on Earth, follows patterns that are best explained by evolution in combination with the movement of tectonic plates over geological time. For example, broad groupings of organisms that had already evolved before the breakup of the supercontinent Pangaea about a million years ago tend to be distributed worldwide. In contrast, broad groupings that evolved after the breakup tend to appear uniquely in smaller regions of Earth. For instance, there are unique groups of plants and animals on northern and southern continents that can be traced to the split of Pangaea into two supercontinents, Eurasia in the north, Gondwana in the south. The evolution of unique species on islands is another example of how evolution and geography intersect. For instance, most of the mammal species in Australia are marsupials, carry young in a pouch, while most mammal species elsewhere in the world are placental, nourish young through a placenta. Australia's marsupial species are very diverse and fill a wide range of ecological roles. Because Australia was isolated by water for millions of years, these species were able to evolve without competition from or exchange with mammal species elsewhere in the world. The marsupials of Australia, Darwin's finches in the Galapagos, and many species on the Hawaiian Islands are unique to their island settings, but have distant relationships to ancestral species on mainlands. This combination of features reflects the processes by which island species evolve. They often arise from mainland ancestors. For example, when a landmass breaks off or a few individuals are blown off course during a storm, and diverge, become increasingly different as they adapt in isolation to the island environment. Fossil record fossils are the preserved remains of previously living organisms or their traces dating from the distant past. The fossil record is not, alas, complete or unbroken. Most organisms never fossilize, and even the organisms that do fossilize are rarely found by humans. Nonetheless, the fossils that humans have collected offer unique insights into evolution over long timescales. How can the age of fossils be determined? First, fossils are often contained in rocks that build up in layers called strata. The strata provide a sort of timeline, with layers near the top being newer and layers near the bottom being older. Fossils found in different strata at the same site can be ordered by their positions, and reference strata with unique features can be used to compare the ages of fossils across locations. In addition, scientists can roughly date fossils using radiometric dating, a process that measures the radioactive decay of certain elements. Fossils document the existence of now extinct species, showing that different organisms have lived on Earth during different periods of the planet's history. They can also help scientists reconstruct the evolutionary histories of present-day species. For instance, some of the best-studied fossils are of the horse lineage. Using these fossils, scientists have been able to reconstruct a large branching of family tree for horses and their now extinct relatives. Changes in the lineage leading to modern-day horses, such as the reduction of toed feet to hooves, may reflect adaptation to changes in the environment. How can the age of fossils be determined? First, fossils are often contained in rocks that build up in layers called strata. The strata provide a sort of timeline, with layers near the top being newer and layers near the bottom being older. Fossils found in different strata at the same site can be ordered by their positions, and reference strata with unique features can be used to compare the ages of fossils across locations. In addition, scientists can roughly date fossils using radiometric dating, a process that measures the radioactive decay of certain elements. Fossils document the existence of now extinct species, showing that different organisms have lived on Earth during different periods of the planet's history. They can also help scientists reconstruct the evolutionary histories of present-day species. For instance, some of the best-studied fossils are of the horse lineage. Using these fossils, scientists have been able to reconstruct a large branching of family tree for horses and their now extinct relatives. Changes in the lineage leading to modern-day horses, such as the reduction of toed feet to hooves, may reflect adaptation to changes in the environment. The emergence of DDT resistance is an example of evolution by natural selection. How would natural selection have worked in this case? More on natural selection. Before DDT was applied, a tiny fraction of mosquitoes in the population would have had naturally occurring gene versions, alleles, that made them resistant to DDT. These versions would have appeared through random mutation or changes in DNA sequence. Without DDT around, the resistant alleles would not have helped mosquitoes survive or reproduce, and might even have been harmful, so they would have remained rare. When DDT spraying began, most of the mosquitoes would have been killed by the pesticide. Which mosquitoes would have survived? For the most part, only the rare individuals that happened to have DDT resistance alleles and thus survived being sprayed with DDT. These surviving mosquitoes would have been able to reproduce and leave offspring. Over generations, more and more DDT-resistant mosquitoes would have been born into the population. That's because resistant parents would have been consistently more likely to survive and reproduce than non-resistant parents, and would have passed their DDT resistance alleles and thus the capacity to survive DDT onto their offspring. Eventually, the mosquito populations would have bounced back to high numbers, but would have been composed largely of DDT-resistant individuals.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time. So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Ago, that life would have been in the form of single-celled organisms, similar to the bacteria of today. Now, I'm not going to discuss how that first cell came to exist, because that's something that is beyond the scope of evolutionary theory. However, there are several ideas out there about how that happened. So if you're interested in learning more about that, you'll want to search for the term abiogenesis. I'm also not going to talk about viruses. You can see how I've placed them off to the side here, because there is currently some disagreement over how exactly they relate to life and whether they evolved before or after bacteria. But what we do know is that life eventually proceeded in 3 different directions. These are called domains, and they are the top classification level, coming before the more well-known kingdom level. So we have bacteria, which includes single-celled organisms that often cause infections in humans, such as cholera or tetanus. Then we have archaea, which are kind of like bacteria and can sometimes be found in really extreme environments, such as super hot or super salty conditions. And then finally, and most important to us, is the eukaryote domain. Eukaryote means true cell and is so labeled because eukaryotes have cells with a nucleus and other inner parts. This originally occurred because of a process known as endosymbiosis, in which several different types of cells came together to form one big cell that was more complex. This likely occurred about 2 billion years ago. From this point, life again proceeded in 3 different directions. First, there were different types of algae that eventually evolved into the plant kingdom. Second, there were various types of mold that evolved into the fungus kingdom. And then third, there were several amoeba-like organisms that evolved into the animal kingdom. All of these simple forms of life are called protists, and in fact, they can actually be divided into a bunch of mini-kingdoms. The fungus kingdom, of course, includes the familiar mushroom, but also things like yeast and the mold that grows on bread. But this chart is mostly concerned with the 2 really big kingdoms, plants and animals. And since we're mostly going to be looking at the animal side, let's quickly look at the plant side before we do so. So from red algae and brown algae eventually came green algae. Plants are green because they have a special part of their cell that other living things do not have. This is called a chloroplast, and it's the part that captures energy from the sun through a process called photosynthesis. From algae came the earliest types of land plants, such as moss. That occurred about 450 million years ago. From there, we get the simplest types of plants with branches and leaves, like ferns. Next, we get a really important development. We get plants that bear seeds and thus are able to replant themselves over longer distances. And it is at this point that the plant kingdom splits into 2 main divisions. We get conifers, which are trees with cones, such as pine trees or cypress trees. And then we get angiosperms, which are trees and plants with flowers. Now, interestingly, flowering plants actually evolved relatively late in Earth's history, only about 150 million years ago. That's halfway through the age of the dinosaurs. And when I say flowering plants, I'm not just talking about things like tulips, roses, and daisies. I'm also talking about the food that we eat, everything from wheat and rice to fruits, vegetables, and even chocolate. All of those things come from flowering plants. Okay, what we really want to focus on though is the animal kingdom. Most people will be surprised to learn that a sea sponge is actually an animal. It might look like a plant, but it's actually comprised of a bunch of simple animal cells bunched together. From this point, what happened is that different cells evolved to perform different types of jobs, such as those involved in movement. So for example, we eventually get jellyfish. A jellyfish is still a very simple creature, but unlike a sea sponge, it can move around. This is because it has nerve cells that send electrical signals to certain parts of its body, causing those body parts to jerk, and thus causing itself to swim. Now keep in mind as we go step by step that each new development along the evolutionary journey took millions, if not tens of millions of years to occur. So none of these changes happened fast. 1,000,000 years is a really long time, and it's really hard for our brains to think using numbers that are that big. So just remember that evolution occurs because the mechanisms involved, such as mutation and natural selection, have really long periods to work with. Okay, so around 550 million years ago, we get what's called the Cambrian explosion. That's when we get animal life starting to go in a bunch of different directions. And it all started with something called bilateria. If you look over at this flatworm here, you'll notice that one side of it mirrors the other side. Well, this is basically true for all animals from this point forward, including humans. That's why we have 2 eyes, 2 arms, 2 legs, and so forth. From here, animals can be divided into 2 categories, protostomes and deuterostomes. Protostomes include the various worm phyla, as well as the phyla Arthropoda and Mollusca. Phylum Arthropoda is comprised of all those creepy crawly things that most humans tend to hate, like spiders and insects, but also larger creatures like lobsters. Phylum Mollusca is comprised of creatures with hard shells like snails and clams, but also some really intelligent sea creatures such as octopi and squid. It is from early deuterostomes that the vertebrates eventually evolved. All vertebrates belong to the phylum Chordata, and they differ from invertebrates, shown in purple, in that they possess a backbone or spine, that long column of bones that runs down the back and protects the spinal cord. The earliest vertebrates were all fish, first jawless fish, and then fish with jaws and teeth. After this point, we then get cartilaginous creatures like sharks, but also for the first time, creatures that have what we consider to be a full skeleton, the bony fish. Bony fish can be divided into 2 main categories. Most fish are ray-finned fish. These include all the familiar types of aquarium fish, such as tetras, guppies, and goldfish, and also all the fish that humans tend to eat, like cod, halibut, and salmon. The lobe-finned fish are quite a bit different. They have downward-facing fins with more muscles in them. Eventually, this feature led to the evolution of certain fish that could prop themselves up in shallow water, like the now extinct Tiktaalik. From a creature similar to Tiktaalik came the tetrapods. Tetra meaning 4 and pod meaning feet. So tetrapods are animals with 4 limbs, which basically includes all of the animals that we tend to think of as being animals. First off, there's the amphibians, like frogs, who live both in the water and on land. But then something evolved called the amniotic sac, which allows some animals to lay their eggs on the land instead of in the water. From this point, animal life proceeded in 2 very different directions. We get the early synapsids that eventually evolved into mammals, and we get the sauropsids that eventually evolved into reptiles and birds. Since we'll be focusing mostly on mammals from this point forward, let's quickly look at the sauropsids before we do so. The 3 main reptile orders that still exist today are the crocodiles, the lizards, and the turtles. Most of the other orders are now extinct, such as the fish-like plesiosaurs and ichthyosaurs, and the bird-like pterosaurs. Although those groups are sometimes called dinosaurs, the term dinosaur technically only refers to these 2 orders here, Saurischia and Ornithischia. Ornithischia, which means bird-hipped, includes dinosaurs like Triceratops and Stegosaurus, whereas Saurischia, which means lizard-hipped, includes the huge, long-necked herbivores called sauropods, as well as the carnivores called theropods. In the theropod group, we get the infamous T-Rex, as well as the raptors. The theropods are also important because it is from that group that birds eventually evolved, one of the earliest being the Archaeopteryx. Now, you might have thought that birds would have evolved from the bird-hipped dinosaurs, but take note that they actually evolved from the lizard-hipped dinosaurs, and in particular from the theropods. The technical term for birds is aves, so the bird class is called the avian class. Note that the ancestors of game birds like chickens and ducks evolved first, and then we get a large group known as the neo-aves, or new birds, where we find all sorts of familiar birds like eagles, pigeons, and crows. Okay, let's now go back to the synapsid line. The earliest synapsids looked a lot like reptiles, sort of like this pelycosaur here. But eventually their skeletal and organ structure evolved along a very different path. Initially, this had a lot to do with movement. If you watch a lizard move, you'll notice that they tend to waddle from side to side, whereas a dog has the ability to run in more of a galloping style. This is a very rough way to explain the initial difference. The first big group of synapsids were the therapsids, and from them evolved the cynodonts. Eventually, the cynodonts developed one of the key features of the mammal class. They became warm-blooded, which meant they also evolved hair or fur in order to regulate their temperature. Of course, the other thing that sets mammals apart is that they possess mammary glands, which they use to feed their young. In fact, this is where the word mammal comes from. One of the earliest types of mammals that still exists today is the duck-billed platypus. They belong to the monotreme order, and most people think they look awfully strange. But most mammals belong to a group called Theria, which simply means beasts. There are two major categories of beasts, marsupials and placentals, and the fact that they evolved differently had a lot to do with geography. For example, today we only find marsupials in the Americas and in Australia. Marsupials, such as the kangaroo or opossum, grow their babies in a pouch, whereas placentals have a placenta and are thus able to grow their babies inside and give birth to them when they are more well developed. I should note that up until the time that modern humans arrived in Australia, there were all sorts of other types of marsupials, types that we don't see today, including marsupials that looked like cats and marsupials that looked like bears. But let's continue with the placental line. Some of the oldest branches of this group eventually lead to elephants, as well as sloths and armadillos. But another branch leads to the Boreoeutheria group, which has two main categories, Laurasiatheria and the supraprimates. Humans are supraprimates, so let's look at Laurasiatheria first. Lots of well-known animal species belong to Laurasiatheria, including all of those that are carnivores. The carnivore order includes bears, dogs, and cats, but also sea creatures such as seals and walruses. Laurasiatheria also includes the ungulates, which are the animals with hooves. Those with an odd number of toes are called perissodactyls and include horses and rhinos. Those with an even number of toes are called artiodactyls and include farm animals like pigs, cows, and sheep, but also animals with antlers like deer. Interestingly, dolphins and whales are also closely related to the artiodactyls. They evolved from land animals that eventually returned to the sea. Okay, let's look at these supraprimates. From the original supraprimates evolved rabbits and rodents, but also the earliest types of actual primates. We tend to associate primates with monkeys, but the primate order also includes animals such as tarsiers and lemurs. Monkeys, as well as apes, belong to a special group of primates called simians. Now, a lot of people use the word monkey and ape interchangeably, but really they are quite different. Apes do not have tails and are generally quite a bit larger and smarter. It is, of course, from the early ape species that humans evolved. Currently, there are only 4 kinds of great apes: orangutans, gorillas, chimpanzees, and humans. But over the last 15 million years, there have been many other kinds as well. It's important to remember that the great apes of today all evolved from these now extinct species, not from each other. So although you may sometimes hear it said that humans evolved from chimpanzees, this is actually not true. Both modern humans and modern chimps evolved from a now extinct common ancestor who lived about 7 million years ago. To us, that creature probably looked more like a chimpanzee than a human, but really they were equally both. From that common ancestor evolved the now extinct genus known as Australopithecus, which included the female known as Lucy, whose skeleton was found in Africa in the 1970s and dated to about 3 million years ago. Australopithecus is thought to be one of the earliest apes to walk primarily on 2 feet. From Australopithecus evolved the genus Homo, which is the genus that modern humans, called Homo sapiens, belong to. Currently, Homo sapiens is the only species in this genus, but over the last 2.5 million years, many other Homo species have come and gone. We know this from the archaeological record, which gets larger and better every year. One of the earliest Homo species was Homo habilis. They were among the first humans to start extensively using tools. Then there was Homo erectus, the African version of which is now called Homo ergaster. They are thought to be the direct ancestor of Homo heidelbergensis, from which came Homo sapiens, our close cousins the Neanderthals, and a recently discovered group known as the Denisovans. You only have to go back around 50,000 years in history to find more than one human species still existing on our planet. At that time, the